In the interest of getting through everything, I'm going to move on to reason 8, which is about avoiding taxonomic uncertainty. And there are two particular examples we've got to show you. One is Acacia acinaceae, or Kakirakanakia. I think David will want me to call it that, right? And Atriplex stipitata. But Linda, would you like to talk about your acacia work? And you'll be wanting your slides, I think.
All right, so species boundaries I think are really important for understanding your ideas around local adaptation and provenance and all those sorts of issues about seed sourcing. And part of it is because things grow across a broad range, and so they might have some different plasticity to different environments, and some might be genetically driven. So this was an example of the variation that's in Acacia acinaceae, which I think now I'm going to have to say Akanakia. That's awesome. This is a couple of peas that have some quite substantial variation. One's in WA. And so we, with Greening Australia, some of you will probably remember when Dave Milson was working at Greening Australia, he got us to look at Acacia acinaceae because they knew there were two ecotypes. It was costing them a lot of money to keep the two ecotypes apart in their seed bank, and so they were asking, could they actually just, collapse all the seed down into one seed lot? So we got 34 populations to assess, and so we did a couple of different assessments. We actually looked at growth, which is the fancy quantitative variation that's basically a growth trial. We did some molecular work using a marker called an AFLP, and then we also did some chromosome variation. And the chromosome variation was quite serendipitous because we just happened to have a colleague from New Zealand who did chromosome counts, and we needed to give him something to do. So we threw this at him and said, have a look at that, not expecting him to find anything. So these were the study sites. They were all collected under seed bank guidelines so that we knew we had good representative populations, and they were nice large ones to choose from. The morphology threw up some interesting results. So on the left is the map. On the right is what's called a principal components analysis, and that's just a way of distributing the populations according to their morphology in two-dimensional space.
What's morphology?
So morphology is the characters that you might, so flower shape, leaf length, height, how things bend from the stem, those sorts of things that taxonomists use to derive species descriptions. Is that okay? Yep. So each one of those letters or numbers represents all that information collapsed down to a single point on this graph. And the first thing we noticed was these two, 5 and 6, fell a long way from the rest of the populations. And so that was a bit of a red flag for us, in that if this was one species with just a little bit of variation, you might expect those to fall with every other population. They didn't. So we went back and looked at them, and they're actually, I'm not a taxonomist, so we called them the hairy ones, because they did have more hairs on them than the others. We went back to the collectors and they went, nope, they fit within the description of this species. So they were considered to be Akanakia. I didn't like that, having them in the analysis, so I took them out because I was a bit more curious. They, to me, are what we call an outlier. We like to take them out and actually reanalyse the data set. So that's what we did. And then we started seeing some other splits between the populations. And so this first split that's diagonal there, the populations on the right of the diagonal actually have 52 chromosomes, and the populations on the left have 26. And this is why we were lucky we had our colleague from New Zealand, because we weren't expecting that at all. We didn't even think there was a chromosomal issue between any of the populations, because they don't look that different. They look a little bit different in terms of this one's a bit prostrate, that one's a bit more upright, but not really that different. So that's a bit of a signal for us that this is a taxonomic complex. Then there's this other split that goes this direction, horizontally, between the 26 chromosomes, and that actually differentiates two types of Akanakia. One is the prostrate one, I have to say that carefully, and then one is the upright form. So this is what we talk about when we need to understand what our taxonomic boundaries are. One of the issues for Australia, of course, is we don't have enough taxonomists, and we've got 21,000 species to work with. However, it gets even more complex than that because there's always an exception to every rule in biology you like to create. There's going to be an exception, and there's this population 8 from South Australia that has 52 chromosomes but looks like this bottom variant here. So, and actually the South Australians don't recognise this necessarily as echinacea. They actually, they actually have their own variant name for it. So it can be a very complex thing. The issue for us around this is that if you mix the 26 and the 52 together, you could potentially, if they breed together, create odd chromosome numbers in the seed. And those seed can often be sterile, and so the population doesn't go forward. It doesn't create new generations. If they become sterile, that's where the population ends. Those 52 chromosome ones were actually found in a very small region around Bendigo, and so our recommendations were don't move that one very far. Don't move that Bendigo form very far from where you find it. Just to give you a little bit more idea that it's not a straightforward, what's provenance, what's the distance, can I tell how far I can move things? When we looked at just those two groups of 26 chromosome populations, genetically, they kind of fall into two groups, but not on a geographic basis. So what we've got is 3 populations in red and 2 populations in blue. And if you really had an easy way to differentiate these on geographic distance, you would expect M, J and A to be the same genetically, but differentiated from U and 3. That's definitely not happened. And then on this one, 4 falls right within the range of what we collected for the second form, but seems to be differentiated genetically. That's basically what that graph on the right's telling you. So it is a complex story, and it's very hard to get rules around these ideas. Yep.
Due to artificial movement, so if that evolves, there is some coming down from different areas and moving in.
It could very well be, because we've got some other studies that we think have got the same thing has happened. Yep. And I just want to flag here, so at the moment, all the translocations in Australia are being reviewed as part of what's called the TSR Hub. And Jen Silcott, who's doing that work, has actually also looked at all the Indigenous movements that she can find the records of. And so what we think is might be our little patch of local stuff may not have been entirely local for a very long time. So just to mention that chromosomal variation does exist, and it can be an issue for choosing local populations. You often can't tell when you look at a plant if it's polyploid. Had the acacia story, Swainsonia sericea, very complex chromosome numbers. Rudidosis has 2 types of chromosome numbers. Microceres is actually a tetraploid, but only has 36 as far as we know across it. And then Eremophila glabra is a great example of 18 chromosomes in Western Australia, 36 as you come across the arid interior, and 54 when you get to the eastern states. So it's got this big range, and it kind of seems to be partitioned up associated with the evolution of that species across the continent. So that's just going to add to your issues, sorry. But it's important you understand that there are these complexities.
Thank you. Now, the other example comes from Neville Walsh. He told me about this, but then he's just relaying it from Ian Sluter, and it regards revegetation, and in fact it's sand mining rehab, and a species called Atriplex stipitata, at least currently that's what it's called. This is just to illustrate what happens, or the point about taxonomy and how well we know how to split things up into different species. On the right-hand side of the screen there, you see a photograph of a spindly, sickly-looking thing with very slender leaves, and that is under present nomenclature Atriplex stipitata. So is the thing at the bottom left. It has broader leaves. It also differs in many other respects. It forms denser colonies. It's dioecious, that is, there are separate male and female plants, which doesn't apply to the well, Neville described it as the gracile form, the slender form on the right. And there are some other differences which mean that if only somebody can sort of do the work on it, they would be described as different species, but they are the same species according to current terminology. So what happened was, when the job came up for sand mine rehabilitation, the person who knew of the thing on the bottom left, and I don't know who they are, but a bloke, decided that that one was going to do a lot better for rehabilitating an area that had been denuded, and so he planted that. He got the seed from South Australia. It was doing really well in his garden, I'm told, and so that was used for the revegetation. Neville now says that they're actually separate species. And there can be numerous other cases that I can think of where this has happened, where people have planted things. For example, some of you from out in the east in Melbourne would be growing things that you probably know as Platylobium formosum, but it's not formosum anymore. And if you'd got Platylobium formosum seed or plants to install in revegetation, they weren't from the local area, they won't be formosum. They will now have a new name. So the fact that something has a name at the present doesn't mean that they're actually the same species as anything else with the same name. So that's just the issue. And it was still an issue which a large number of people identified as an issue for choosing local provenance, because at least if you choose local provenance, it won't matter if somebody changes the name of it subsequently, but if you choose something that isn't local, it might then be discovered not to be the local species. And I'll make a prediction, Lamandra longifolia might end up in that category. Now, Jeff Carr would like to say something. Do you want the microphone here? Oh, here it comes. Now, as I mentioned, I was the original Vice President of IFA. Jeff Carr was the original President of IFA.
Jeff Carr, taxonomy is a hugely hoary issue and we could talk about it for the rest of the week, but I'm suggesting that there's a big question in people's minds: how do you find out what this thing is? This thing that I'm collecting seeds from. And in some locations there are two different forms which to me clearly connote different taxa. For example, in the chenopods, Einadia nutans, there's a prostrate thing that forms a tight mat, another one that can grow next to it which grows half a metre or more high. The same thing in Enchilina tomentosa, the barrier saltbush. There's a prostrate thing. Next to it you can have a twiggy thing that grows about a metre high, much hairier. Clearly they're very different things. In Troglocan procuring that we were talking about before, there are at least 3 different taxa, I believe, in procuring in the Melbourne region. So the big question is, how do you find out? And I'm not proposing to answer this question here, but the question does need answering, and a corollary problem is that I see now frequently plants in nurseries under the wrong name. For example, Dianella longifolia, so-called, which is, we were told by the literature we've known for a long time that we had longifolia around here. Well, there are 3 different things in that group here, but the stuff that's being flogged in at least 2 Melbourne Indigenous nurseries is actually Dianella calicarpa, which occurs no closer than about 200 kilometres to Melbourne. And so it goes. There are 2 Dianella revoluta, there are 3 Dianella revoluta things around Melbourne. But the big question is, how do we find out? Because a lot of this data is unpublished. It's in the crania of a few smartasses. Or in some cases, there's really good literature. That's all I want to say at the moment.
A very helpful contribution. Thank you, Jeff. Anything else that anybody wants to say? Well, more and more questions. My mind is boggling. I'm thinking that anybody who isn't confused simply doesn't understand the situation. I see that there is a question from Naomi Sunner up the back there. We'll run the microphone down. Warren can be very quick, up near the back on the left.
I was just wondering, where is the best spot for for people working in the field to find that information? And Jeff just mentioned that some of it's not published, but where would be the first point of call for people to go to? Sometimes the online Flora Vic isn't up to date.
Well, even if it's up to date, up to date with regard to the academic taxonomy, it won't tell you informal recognitions of Dianella, say longifolia group being split up into these things within the Melbourne area, because the scientific work hasn't all been published and cut and dried.
But if it has been, where would be the best point of call for practitioners to go to first up?
Anyone got an answer? Okay, for identification, Liz James is offering the services of the people in the room behind us, or behind me, in front of you. Ring and make an appointment, 9252, anyway. You can bring specimens here for those sorts of purposes, although for commercial stuff you'll typically be asked to pay. And for Jeff Carr has just given you the benefit of his very in-depth expertise on those particular groups and many others as well, like couriers and so on. But you won't necessarily get that if you just go to any other person. There is no simple repository. You better be a member of IFA, I think, Naomi.